Welcome to Phylum Porifera, the pore bearers. Phylum Porifera, the sponges. A few examples: stovepipe sponges, red volcano sponge, yellow tube sponge. Phylum Porifera, 5,000 living species, 3 distinct groups classified by skeleton type. Hexactinellida, the glass or silica sponges. Demospongia, spongin or collagen sponges. Calcarea, calcareous sponges. They're the most primitive of all animals. Identifying characteristics of the members of Phylum Porifera: mostly asymmetric, some exhibit a radial symmetry. Sessile, anchored to the ocean floor as an adult, although larvae can swim. Found mainly in marine habitats but can be freshwater. Non-coelomates cannot possess any true body systems, although they do have highly specialized cells that perform many functions. But they do possess a gastrovascular cavity, or GVC. They are filter feeders through pores and special cells that line the GVC. They are hermaphroditic. They have a unique skeleton and cell structure. What do you think the gastrovascular cavity is for based on its name? Gastro equals to do with digesting food and absorbing nutrients. Vascular is to do with circulation. It's a primitive circulatory system that moves those nutrients around. The structure of a sponge: it has water that flows out through osculum. The water gets dragged into entrances of the filter chambers through the ostia, singular ostium. There is an enlarged area below. There's an ostium, an atrium. The water flows in through many ostia. Sponge skeleton and feeding. The skeleton is made up of spicules, composed of silica, spongin or calcium, and/or a protein mesh of spongin. Structural wall consists of 3 layers: the epidermis or outer layer of pinacocyte cells with porocytes. These make up ostia or pores. Middle layer of amoeboid cells, and an inner layer of choanocytes or collar cells. The porocytes allow water to enter. Amoeboid cells transport nutrients, produce spicules, form sex cells. Choanocytes engulf and digest food, vacuoles. Have flagella to pull water through pores and circulate it through sponge. Filaments trap food particles. Waste and water are purged through the main pore or osculum. The flow of water is unidirectional. In the enlargement of a collar cell, you can see that there's independent beating of many flagella which force water out of the large osculum and pull water in through the ostia. Particles of food are drawn into the collar to become trapped and then engulfed by the cell body and digested in the food vacuoles. In the second picture, in figure A, you can see yellow tube sponge, or Aplicina fistularis. And in figure B, you can see the sponge organization. Water goes in through the pores. Water goes out through the osculum. There's a central cavity. The sponge wall consists of the amoeboid cell, the porocytes, the epidural cell, the spicules, the flagellum, nucleus, collar, flagellar fibrils, collar cell, choanocyte, and amoeboid cells. With regard to diet and feeding, sponges are filter feeders. This means they eat bacteria, protists, and sometimes small crustaceans. For example, the larval stage of shrimp. Essentially, collar cells, or choanocytes, collect and digest food, then bring it to amoebocytes, or the amoeboid cells, for distribution of nutrients. Sponges take in oxygen as water passes through the body through diffusion. In the area of reproduction, they are hermaphrodites. In other words, they make eggs and sperm. They use asexual reproduction, which occurs via budding, so they produce internal buds that can grow into new sponges when the conditions are more favorable. Buds are called gemmules and are protected by a layer of spicules. They can remain dormant if necessary. Regeneration, or growth of a whole organism, can occur from a fragment. Archaeocytes are another name for amoebocytes or amoeboid cells. They have, in the diagram as you can see, micropyle, inner membrane, archaeocytes, and spicules. In the sexual reproduction of sponges, egg and sperm are produced by meiosis. Sperm are released into the water. Sperm enters pores of another sponge of the same species and swims up the collar cells to fertilize an egg. free-swimming larvae released through osculum. Whether by asexual or sexual reproduction, sponges have poor reproductive odds. Larvae find a hard surface to settle on and undergo metamorphosis to become a sessile adult sponge. Sessile means that it's fixed to the bottom. In the life cycle of a sponge, the gametes are sperm and egg. They have dividing cells, spongocoel. Larva are released, the flagella moves, and a new sponge develops. The ecological importance of sponges. They provide shelter and food for other ocean creatures, can release chemicals that help to break up old shells. They form symbiotic relationships with bacteria and plant-like protists. Because sponges are filter feeders, they are very sensitive to water pollution, and it will quickly clog their pores. A lack of sponges in a certain area is a good indication that there is pollution in the area and more ocean life is potentially at risk. For example, they're like the canary in a coal mine. Sea crab living inside a fluorescent sponge is in the picture at the top, and in the picture below, these holes were caused by a boring sponge. The economical importance of sponges. At one time, there was a market for sponges to be used in the bathroom and the kitchen because of their ability to absorb water. However, synthetic sponges have replaced this industry. Sorry about that. I imagine that's kind of a cool fact that your teacher would include. Super cool sponge facts. Each species of sponge somehow knows exactly which day of the year the other members of its species will release their gametes into the water. This is how sponges are able to cross-fertilize. The loofah sponge, sold in many stores, is not actually a sponge at all. It's the inside of a plant known as a gourd.